Right, so thank you first of all for the opportunity to talk here in the VarI session. I'm going to be talking about a framework which is using some of the similar concepts to what Alpha Missense just talked about. But I'm going to try and introduce a couple of different tack, if you want, on the whole problem. Because at the moment, so when we understand proteins, we typically have the sequences, of course, and we also have structures. Yeah, and there's many structures available, and this, of course, has been an enormously valuable resource. But I'm trained as a chemist, and proteins are incredibly complex molecules, the way that they behave and what they can do. So their energy landscape is also incredibly complex. So this is, of course, a vast oversimplification of what it actually looks like. But the key principles remain that, you have thermodynamically stable states, right, which have low energy, like here, and you might have some other ones. And then kinetics is about moving between these states, right? Can it move between these thermodynamically stable states? Now, when you think about the data that AlphaFold has been trained on, it's typically X-ray crystallography diffraction structures. So this is like proteins at cryogenic temperatures in a crystal lattice. Yeah, so this is not really what they're doing in solution. And AlphaFold, of course, is very good at predicting this. And essentially, these are the low energy states that you have for a protein. And I would also like to point out here, of course, that this has only been possible because of decades of public data curation. That's always an important point to make. But what can also happen is that your environment changes. You might have a variant or a mutation, or you might have a personalization modification. And this might change your energy landscape, which means that another state might become more stable, right? Or the whole energy landscape even changes. And this is much trickier to address, of course, and to capture. And then on top of that, this all happens within the context of the cells or the proteins interaction. So the whole picture is very complex when you look at the protein in its in vivo environment. So what we've been thinking about is function and what I would call biophysical conservation, which I will introduce later. If you think about evolution, right, we have extant sequences, structures, environments that these proteins operate in. These come from ancestral proteins somewhere down the line. And essentially, what we assume, of course, is that the protein function is conserved. You could sort of think of it a bit like, I'm not sure if you can see that, like an old bike. Has now evolved into like race bikes and like, electric fat bikes and all these things, right? But essentially, they're still bikes. Yeah. So this is sort of, I guess, the function remains the same. Now, if you then go to the biophysical level, there must also have been constraints on how the protein behaves in order to function in the correct way. The structure is one example of that, but there's more to it. And this is then also, in principle, conserved in this protein in order for it to keep on functioning. That doesn't come out well on the screen. It's good on mine. Anyway, so that's the observations that we have at the moment. And what you can now sort of think about is that you might see the gear and the crank and the chain on these pictures, but you don't know that they need to move in order to propel the bike forward, right? So there are these like hidden features, if you want, in proteins that enable them to function. In the case of the bike, that would be the gear. And so there's this hidden layer, if you want, of these protein biophysical semantics, as I would call it. Now, the protein behavior necessary for the function is conserved, right? So, and this is, of course, often directly related to the fold, but also important are these hidden biophysical features. That's the point, I think. So things like folding pathway, conformational states, dynamics that are not captured directly by sequence or structure in its traditional form. So you have the sequence, you have the structure, and then what you can sort of say is that what we try and look at, is the local interactions in the protein sequence, so the emergent behavior, if you want, what your protein is capable of in a way. So it's more of a probabilistic view of proteins, like their capacity to do different things, if you want. Now, we have been developing, and this is now 10 years back already that we started to do this with biophysical predictions. There's a twist to these predictions compared to most of them. They're based on data for proteins in solution from NMR. So you actually can include many different types of proteins in your data set. You can then train it. It's a simple linear regression, in fact. And then you can predict just with a sequence, single sequence, these characteristics. And this has been out for a while. So you have basically dynamics and conformation. We call these biophysical features. And then you also have predictors that use the combined behavior to do things like predicting early folding and disorder and things like that. So where you have the combination of these different features. Now, what you can do is, say you have a sequence. This is a tin barrel, well-known structure, well-known secondary structure as well, of course. So you can run, no, this is not working. It's not showing up on my Mac. It is. Yeah, okay. So I'm gonna skip this or try and do this more quickly. So essentially, you have a prediction, right? A single one. If you then have a multiple sequence alignment, you can run these single sequence predictions for all your different sequences, and then you get like, yeah, unfortunately I can't show it here. You get all these predictions on top of each other. And what you can now do is not look at the individual sequence, but look for each column in your multiple sequence alignment what the distribution is of your predicted features. So for example, backbone dynamics. Yeah. And then here, yeah, now it's showing up so that you get something like this, right? So you get all these different predictions on top of each other. And here you see the sequence conservation. And there is, in fact, the correlation between sequence conservation and the variation in your biophysical features is very bad. So in effect, you can have regions that are very variable, but are biophysically very conserved in their behavior. That's the key point, essentially. Because it's not really the sequence that matters, it's the behavior of the protein. That's the thinking. Okay, so if you blow this up then, yeah, what you can then do is make this into a distribution, essentially. And here again, there's a grayscale around it that's sort of box plot-wise. This is the median. And you should imagine that you have a range around it for the box plot itself. What you can then see is that, for example, here for the alpha helices, is that the patterns that you get, and this is, again, only the median, for alpha helices can actually be quite different. Yeah. So here, for example, yeah, this alpha helix has this big peak in the middle, and this one has a very different pattern. Now, they're both alpha helices in the structure, but there is something in these biophysical features that is different about them. I have no idea why, but it's there. That's a signal that comes out of this analysis. So you can do this then also for other things, like here as a final example, you have the beta strands here. What you can see is that this is now the beta sheet propensity, not the backbone dynamics. What you can see is for some of these beta sheets, they have a very strong local propensity to form beta strands. Others do not. So probably these are from later down the line in the folding path where they're not so essential to get the fold right. And we've actually shown this. So in fact, then you have these biophysical annotations, you've got a sequence. So you can then do, we have like about 7 or 8 of these. What you can do is basically replace your amino acid by these biophysical annotations. Yeah, so you get instead of an amino acid, you have a feature vector. And this is not the same for the same amino acid because it has a different position in the sequence, and therefore also its characteristics will be different essentially. So you can use a local sequence-dependent biophysical signature, at least in the conservation, and we've already used this in different applications to show that you can distinguish between proteins, design proteins that fold correctly or not, because they capture something about the folding pathway, essentially, what's really important for protein to fold correctly. Now, and then came, of course, the protein language models, right? And I'm going to go over this very quickly. You need enormous amounts of data, neural networks. I'm not going to get through this. Now you have 1024 features per amino acid or other ones. These are not directly human interpretable, as opposed to what I showed you. They have no real physical meaning, but they do essentially capture the semantics of natural protein sequences at a statistical level, these features that you get out. Now, Konstantina started working on this, and she developed this Delta to Deep platform, where we basically thought, why not try the same principle on these protein language models, right? So you have the protein language model, you've got evolutionary information. I'm not going to get into the details because otherwise I will run out of time. And then what we do essentially is we slap a Gaussian mixture model on it, which captures the variation of the protein language model features based on the specific evolutionary information for that protein family, essentially. So this should then capture the boundaries of what that protein is allowed to do and what not, essentially, right? This is millions of years of testing. And then we have it doesn't, so I'm not going to get into the detail again, but essentially you look at the wild type and then a mutant, and then you measure some distances. You pour them into a feature set, and then you can train predictors. And what we've done first is to try it on cancer driver prediction. So cancer has a lot of, or cancer cells have a lot of mutations, both driver mutations that progress the cancer and pathogenic mutations that randomly occur. Distinguishing between these is quite difficult. We developed a training set. Again, going back to what Yun said, this is like 4 lines, but it's a lot of work to get something balanced. So what we did instead is really use a lot of genes and then try and balance the driver and passenger mutations within those genes. So have a different spread of them. Then the first thing that, of course, we tested is, does this actually add something? And what you see here is, if the mouse moves, yeah, so these are just the vanilla language models using a similar approach. And then this is delta to deep using the evolutionary information, and the performance really increases because it captures essential features of those proteins, essentially. That's the idea. It also performs well compared to other predictors. So this is 2 test sets, the DRGN set, which is sort of tries to be gene non-specific, and then the ClinVar test set, which I think is the one that Yun talked about. You can see alpha missense, of course, does really well. Delta to deep also does really well, especially on the set that is more gene agnostic. And then there's some other methods that use context, like Duogen, that also was developed in our lab years back, that also try and take the protein context into account. So it works, that's nice. So then when you start to look into what you can do with this kind of framework, this is now TP43, 53 at least, yeah. You can just run, of course, the predictor on all different mutations, and then you get this pattern similar to what Yun talked about. Most of them are in DNA binding domain, not surprisingly. Here, the bad ones and the red deleterious ones. But also what's interesting about these protein language models is they do detect long-range connections, as is already known, otherwise you wouldn't be able to predict structure with it. So here you see the distance from the mutation site, and you actually do get long-range contacts that are also taken into account by the model, because these will also move away from the Gaussian distribution in those positions, right? These turn out to be also residues that are close to each other in the sequence. Of course, that's not so new. What is also interesting is that you can use the confidences. So there you look at essentially the distribution, and you try and assess how much of the, how normal your mutation is, essentially, compared to the full distribution. And then you can see that if you take the more confidence predictions that, like here, that's the correctly predicted benign experimental, the incorrectly predicted, correctly predicted, pathogenic, and incorrectly. You can see that the ones that we correctly predict are also more confidently predicted. So you can use this information from the distributions to also make an assessment on how much you can rely on a specific prediction, which is nice, because you have distributions. So then, okay, yeah, and then going back to the biophysics, because then you want to interpret this, of course, because you can't really interpret protein language model features. Here you can see this is the early folding distribution that I was talking about earlier. For a benign one here, you all basically almost don't see a difference. And here a pathogenic one, if I can find the mouse pointer, there it is. You actually get an increase in the early folding signal in a specific position here. So this is likely then why this protein might misbehave. And you can then, of course, because you have this for all these different predicted mutants, you can do this at a statistical level. And then, for example, you can see that for pathogenic mutations, so the drivers, apparently in P53 at least, you get a lot of the pathogenic mutations make it fold more. This is essentially the thing, or like increase its early folding, which is interesting because, of course, it has to be quite dynamic, and this seems to then actually mess up its function. Whereas for the benign ones, you really don't see any difference at all. You can also use this for backbone dynamics and other ones, but it allows you to sort of make an interpretation of what the reasons might be. Now, this is a generic framework. I talked about the cancer driver passenger. You can do this for all kinds of other problems, which we're trying now. For example, in this mega scale stability information set, so 8 million mutations, lots of sequences, stability measurements, a big data set. D2D here does, just without any specific training, does as well as other specifically trained models. You can also even train it protein specific, and then it gets even better, which is cheating a bit, but then you better capture what is essential for a specific protein. Okay, so functional constraints, as I think I've shown, can be captured by this evolution-informed distributions of amino acids, feature vectors that encompass hidden protein family requirements. The multiple sequence alignment is crucial. We've also developed the methods to do this better. We used MMC2. In the beginning, it's applicable to a range of challenges. These distributions enable you to actually look at confidence and significance scores, which is interesting. You could also, for example, compare two different families to each other. And it's also suitable for multiple variants because it doesn't really matter, and also captures epistatic effects. I would like to acknowledge especially Constantina, who did the bulk of this work, and then Daniela and Gabriela were involved in the previous biophysical work. Thank you for your attention.
Actually, I can have a question.
Yes.
Sure. I just want to make a suggestion. So
Yeah.
I believe your method is trying to predict driver mutations or driver genes in cancer.
Mutations.
Both, or?
Well, you could run it on all different.
So these are somatic mutations. And I noticed that when you compared with other methods, all the methods that you used are predictors of germline variants, which have very different characteristics from driver mutations. So you might be interested. There are computational methods designed to predict the impact of driver mutations. And I'm glad to talk to you about this offline.
Yeah, thank you. Questions?
Great talk. With the TP53 analysis with D2D, have you looked at the difference in folding, this correlation with the functional assays that are available?
No, we've not gone into that kind of detail. No, this is it's just to illustrate the concept more now. Okay, yeah, but if you have any good data sets, we can definitely look at it.
Yeah, that's one of the ones. But
okay,
Rich,
thank you. All right.
Thank you.